Hey guys, it's Professor Tiffany Martin with Medical Microbiology, and we are covering chapter 13, which is the chapter on viruses. We're just getting started on that. So, viruses. Why do we want to know about viruses? Well, again, the more we know about the pathogens and how they attack us, if you will, the better we can do for our patients. So, we need to learn more about the structure of viruses so that we can protect ourselves. And this is obviously apparent right now because we are dealing with the after effects of SARS-CoV-2, which is the virus that causes the disease COVID-19. So we're definitely interested. So just take a second and I want you to think about how many viruses that you know of that causes diseases or a disease that's caused by a virus. So just take a second and do it. All right, how'd you do? If you didn't get very many, start thinking about your vaccines, right? What about tuberculosis? We have well, we don't have that's the bacteria. Okay, start this over. Maybe I should just start the tape over. No, that's okay. I made mistakes. Not tuberculosis, but what about chickenpox? What about viruses, vaccines for MMR, right? Measles, mumps, rubella, all viruses. What about your what other vaccines do we have? The pneumonia vaccine. There's a vaccine for that. That's part bacterial, part viruses. Those are that. What else? Mononucleosis. Epstein-Barr virus is one. Herpes, HIV, all viruses. What else? What else? Okay, hopefully you did better than I did. Cytomegaloviruses, that's another one. So anyway, so there's a lot of viruses out there that cause disease, a lot of your case studies caused by viruses. So again, the more we know about them, the better we can do. So let's talk about what a virus is. Okay, before we do that, how do we know viruses exist? Well, we can see them. We have an electron microscope that we can actually see them, so we know they exist. The electron microscope wasn't actually invented until the 1930s. How do we know about them before then? If we go back into history, if you remember chapter 1, remember that they were all looking for, after Pasteur put forth the germ theory. If bacteria, these microorganisms that Leeuwenhoek saw, if these microorganisms can spoil wine, can they spoil people? So everybody started looking for these microorganisms and their connection to different diseases at the time period. And that led to the field of studying etiology, what causes these diseases. So if you remember right, it was Robert Koch was the first person to match up Bacillus anthracis, a microorganism, a bacteria, to the disease anthrax. So took a lot of good research and scientific method on his part. He put forth the Koch's postulates after that, but it also took some luck because if he had been studying influenza, do you think he would have would he been able to figure it out? No, influenza, the flu, is caused by a virus. And so he would not have been able to figure that out because he would not have been able to culture it outside. Looking at bacterial diseases, there was a lot of matching that was taking place, but viral diseases were a little bit tougher. So there was some investigators in 1892, and again, they were looking for the cause of a disease. Now, this guy, Ivanovsky, and both of these guys actually were botanists. They actually studied plants. They weren't physicians, they were botanists, and there was a crop, tobacco, which was a big cash crop, just like the wine industry. And there was a disease that infected tobacco plants. And it was called tobacco mosaic disease, and it caused the leaves of the tobacco plant to turn brown and fall off, and the plant would be sick and die. So obviously that affects profits. That's a big deal. Now they knew this disease was infectious because they could take an infected plant that had this disease and they could rub the leaves on a healthy plant, and then the healthy plant would get the disease. So they knew that it could be passed from plant to plant. It wasn't something in the DNA or anything else at that time. They knew it was infectious. It could be passed from plant to plant. So they were looking for the bacteria, the microorganism that caused it, because so many of these were being matched up at this time period. So Ivanovsky was working in his lab, and he was looking using Koch's postulates, and he was trying, and he could not find a bacteria that caused this disease. And finally, he took through, because he knew he could take the leaves of the plant and he could grind them up into a fluid, into a liquid, and that liquid was still infectious. And so he started passing it through filters and taking things out of the liquid, trying to find where that microorganism was that was infecting it. So he passed it through these filters, and it remained infectious, and it remained infectious. So finally, he passed this fluid that was made from crushing up these sick tobacco plants. He passed this fluid through a porcelain filter. Now, porcelain was a porous material, but the pores in the porcelain were so small that bacteria could not pass through it. You could still get liquid to pass through porcelain, but not a cell, so not any bacteria. So he finally passed it through something that wouldn't let anything through except just a little bit of liquid, and that filtrate, that liquid, was still infectious. So he came to the conclusion there must be something smaller than bacteria that was causing this tobacco mosaic disease. And in fact, he found that there was something very small causing this. His conclusion, Ivanovsky's conclusion, was that this must be a really small bacteria that they could not see under the microscope. So that was his conclusion. Now, there was another scientist in another country working on the same project, and he heard of Ivanovsky's experiments, and he decided to repeat them in his lab. So again, two separate places, two different investigators. He wanted to repeat this experiment, which is a good thing. Anytime you can repeat something and get the same results, that leads validity to those conclusions. So Bergenic was decided to repeat that experiment. So he passed it through a porcelain filter. He got the same results. The fluid was still contagious, even though none of the cells made it through. Now, he was a little bit more forward thinking, and he said, I don't think this is a bacteria. I don't think this is a cell. I think there's something smaller than a cell that we have not seen before that's causing this tobacco mosaic disease. So because he felt it was something new, you get to name it. If you find something new, you get to name it. And so he named it Contagium vividium fluidium, after the contagious fluids, and vividium means poison from that. It got shortened because that was a mouthful to a filterable virus, and then eventually we just called it a virus. So he was the first person to realize that he was dealing with something new and give it a name. So before they could see a virus, they knew they were there based on its actions and what it could do. All right, so again, more work was going into it. In the 1930s, there was a biochemist that ended up purifying this virus and isolating it. And what was interesting about this is that when he isolated it, it crystallized. And cells, if you purify the cells, take out all the water, then the cells just die, right? You don't get crystals formed. Things that form crystals are your minerals, salts, minerals, geodes, those types of things. Inorganic material crystallizes. So this was kind of interesting, and they're like, again, I don't think this is a cell. I think this is something new. The electron microscope was invented, I said the 30s. It was invented in the 1940s and allowed us to, instead of passing light and magnifying with light, we were able to pass electrons through an organism and then magnify it and see that electron pattern. This is how an electron microscope works. It means that we can magnify things much smaller than ever before. And so we got our first pictures of a virus. So this is a couple of different types of electron microscopes. This is a 3D image and some different viruses. This is both, I think it's smallpox. This one is Ebola. It's a different type of shape to it. All right, so finally in the 1940s, we were able to see a virus. And so we knew it was there around 1900, but we could actually see it in the 1940s, but we're still trying to figure out what it is. Is this a living organism? So now that we can see it, we can explore it even further. All right, so the question became, what is a virus? Is this something that is alive or not? And so the way we define living organisms in biology is organisms that can grow. They have to exhibit some certain characteristics. So growth is one. Reproduction. being able to respond to the environment, being able to take in nutrients and build things, so metabolism. So all of these are things that we use to define life. Viruses, if I were to ask you, viruses, living or non-living, what would you say? Viruses, alive or not alive? Okay, so if they're non-living, are they the same as a table? Like a table's definitely non-living. What about viruses? It's a little harder to say they're not living when they have so many when they can infect us, so they can reproduce. Viruses are considered at the cusp of life. They do have some characteristics similar to living things, but they don't have all of the characteristics that biology has decided define a living organism. So viruses are not currently considered alive, but they are kind of at the cusp of life. So let's look at what they can and cannot do. They are made up of nucleic acids, which is like living things. But you remember that central dogma, DNA to RNA to protein? Yeah, viruses don't do that. Viruses can be DNA and go to RNA to protein, but viruses can also just be RNA and not have any DNA. Viruses contain DNA, but they may not contain anything else. So they don't contain that, not until they get into a host. So that's different. They can reproduce, but not on their own. They have to be within a host. They're classified as an obligate intracellular parasite, so they cannot reproduce on their own. Living things respond to stimuli, but the viruses don't do that. Viruses can evolve because they're made up of nucleic acids, but then they don't have a cell structure. They don't have that phospholipid bilayer that we says make up a cell. They don't grow. They don't undergo metabolism. So again, cusp of life, but they are not technically living organisms. All right, so what are they? If there's no cell membrane, what are they? They are considered the smallest infectious agent. Whoops. So I am going to have you put kind of a asterisk about that. We're actually going to, at the very end of this chapter, talk about something that's even smaller than viruses, but they are super tiny. They are classified as ultra microscopic. We can't see them under light microscopes. We have to have an electron microscope. Comparison, if I took an average bacteria cell, and I don't know if you've really understood how small a bacterial cell is, but we have to magnify if you go back to microscopy, we have to magnify a bacteria cell 1,000 times to see those little bitty dots that you saw on the microscope for under that. So that's a lot of magnification, 1,000 times just to get those little bitty dots. You can't really see much other than a circle, you know, if it's a coccus type of shape. You can put 2,000 viruses, bacteriophage, within that little bitty dot that's a bacteria cell. If you talk about a eukaryotic cell, they're much bigger. 50 million of polioviruses, which granted are one of the smallest viruses, but still 50 million can fit inside a human cell. Let me show you some other comparisons. All right, so red blood cell. This picture, this diagram is showing you the size of a red blood cell if we were to magnify it. Now, if you think about a red blood cell, my A and P people, right? Circulatory system, right? Red blood cells going through that. The smallest tube that a red blood cell has to go through, a capillary tube. The size of a capillary will fit one red blood cell at a time. So they go through single file. The very tiniest of parts of our circulatory system is a capillary, and the red blood cells go through one at a time. So that's a size difference. So get that into your mind. And then let's go back to our pictures. This is a red blood cell. So this is the smallest tubes, our capillaries, and they'll fit through one at a time. If I compare this eukaryotic red blood cell to a prokaryotic bacteria, you can see the size difference. This bacteria, oh, it's flagella. What's that arrangement? Oh, yeah, I hope you said paratrichus, right? Flagellum all over the place. Flagella all over the place. So this is a prokaryotic cell. And remember on that microscope, they just look like little dots, little bitty tiny ovals or circles. So that's a prokaryotic cell. If you magnified 1,000 times, you saw what those looked like. These little bitty dots that you can't even see, let's magnify this. So now I've got my prokaryotic cell. These are viruses. Can you see the size difference? If I were to magnify these viruses, now we can get a little more detail. And that poliovirus, 30 nanometers. This is how small that is. A red blood cell by comparison is 10,000 nanometers. So that's quite a bit of a difference in size comparison. So these are really tiny. So think about one virus that gets inside the human body. It can move through, it doesn't move on its own, no flagellum, unlike this prokaryote with lots of flagella here. Doesn't move on its own. It's not living if it's not inside a cell. It's not even living inside a cell. It's not living. It can't move, but it is carried based on currents. So think about it if it gets into your circulatory system. Those currents are air currents. If we breathe that in, so somebody right next to you is coughing and coughs up these respiratory droplets. These droplets have virus particles within it. You inhale them through air currents, gets inside your body, gets into your circulatory system. Very easy for them to move through just based on currents in the body. So it really can get in. It's very difficult to get rid of it. So you can see how one virus gets in. Once it finds a cell that it can invade, replicates, we'll talk about that in a minute, it can be a big problem. Okay, so if this virus, we've said it's really, really small. If it's made up of things, we need to figure out what it's made up of. So I think I'll stop this intro video here, and then we'll figure out, okay, so what is a virus made up of? If it doesn't have a cell membrane, what does it have? So join me in the next video, and we'll talk about that. Thanks.